part 3. Okay, so part 3 includes how the backbone of DNA is formed, how the bases are attached to the ribose sugar, how these two strands are anti-parallel and remain in a perfect conformation. So let's see. There are two ends to DNA denoted as 5' phosphate end and 3' hydroxyl end, which runs in opposite direction, means on one side one strand, if it is 5', on another side it is differently present end, that is 3' hydroxyl end. If there is 3' hydroxyl end, on the another side is there is a different end, that is 5' phosphate end. So what is phosphate actually? It is H3PO4. It is H3PO4, as we studied in the past two parts. Okay, so phosphate is nothing but H3PO4. Hydroxyl group is of ribose sugar. Is of which sugar? Ribose sugar. So let's see how the backbone forms. If there is phosphate group, on one side, there must be fifth carbon of sugar. We can denote it as, this is the sugar. Okay, which carbon of sugar? Fifth carbon of sugar. Let's see how. This is how you can denote the backbone of DNA. Okay, you know the structure of deoxyribose sugar. So let's see how this backbone forms. Phosphate sugar. Likewise, the conformation or structure continues. Phosphate sugar, then again phosphate and sugar. Likewise, oxygen, the phosphate is joined here, phosphate, again sugar, then again phosphate, then again sugar. Likewise, the thing continues. The distance remains the same. But it must be same. Okay, so while drawing, remember that the distance between the ribose sugar molecules must be the same. See, there is an equal distance. So this is 3' OH group of deoxyribose sugar, and this is 5' end, which is phosphate end, which is nothing but H3PO4. So this structure, repeating phosphate group along with the repeating deoxyribose sugar, is known as backbone of DNA. So this structure is known as, this whole structure, it is known as sugar phosphate backbone. Which backbone? Sugar phosphate backbone of DNA. Sugar phosphate backbone of DNA, because of which the DNA gets strongness. It is strengthened by this sugar phosphate backbone, and this phosphate group carries the negative charge. So that's why the DNA is negatively charged. DNA is having negative charge, is having a negative charge due to phosphate group. A negative charge due to phosphate group. So DNA is having negative charge due to phosphate group. The phosphate groups are negatively charged. That's why DNA is having a negative charge. Now let's go further. If you want to write it down, write it down. Now let's go further. You can pause it and see. Okay, so I will go further. What are these bonds between a sugar and phosphate? These bonds are known as phosphodiester bonds. These bonds are known as phosphodiester bonds. So what are these? These are phosphodiester bonds. See, this is ester bond, first ester bond. This is another ester bond. These two are ester bonds, first and second. So the linkage is known as phosphodiester bond linkage. What is this linkage? Phosphodiester bond linkage. See, there are two bonds on both sides of sugar. At fifth carbon, here it's fifth carbon, here it's third carbon. Here is the fifth carbon of sugar, here is the third carbon of sugar. So there are two phosphodiester bonds attached to the sugar on both sides. A phosphate is having attached with two sugars on both sides. So these bonds are known as phosphodiester bonds. Okay, or you can also take this bond, both sides of phosphate group, phosphodiester bonds. Okay, two bonds are phosphodiester bonds. So you can pause and see how the phosphodiester bond is there. So these bonds are phosphodiester bonds. Which bonds? Phosphodiester bonds or linkage. Then how these sugars joins the bases? Let's see. These sugars joins the bases with the help of a bond known as beta N glycosidic bond. This bond, which joins the adenine with deoxyribose sugar, is known as beta N glycosidic bond. Beta N glycosidic bond or linkage. So the nitrogenous base is joined with deoxyribose sugar with the help of a bond. We call it as beta N glycosidic bond or linkage. Beta N glycosidic bond or linkage. Now let's see how the bases are joined together of one strand with another. Because the both strands running are in opposite direction, let's see how to show it. So here the sugar must be shown opposite like this. O is on downside. The O is on downside. Here there is a phosphate. Here there is a phosphate. Then again another sugar molecule will come. Okay, likewise. Again the ring oxygen is on the downside. As you can see, the ring oxygen is on a downside. Okay, then another phosphate is there, then another sugar is there. Likewise the thing continues. Let's see how. Like this. So as you can see, I will show you. Let me draw first. This is how two strands of DNA run in opposite direction. Okay, so here is 5 prime phosphate. Now you can see clearly how the two strands run. This is running in this direction, 5 prime to 3 prime, and this strand is running in opposite direction, 3 prime to 5 prime. So they attach with the bases by using the first carbon. So whatever the strand, left side or right side strand, first carbon of sugar is always joined with bases. Suppose on this strand sugar is attached with adenine. On another strand there must be thymine, because it is a rule. Adenine must join with thymine, or thymine must join with adenine, or guanine must join with cytosine, or cytosine must join with the guanine. So there are double bonds. Here if we consider guanine, there must be on another side first carbon of deoxyribose sugar, cytosine. How many bonds will be there? 3. If the pairing is GC, 3 hydrogen bonds will be there. If the pairing is AT, how many hydrogen bonds will be there? 2 hydrogen bonds will be there. Then again if we take T here first, so T is also joining or will join with A with the help of 2 hydrogen bonds, and this A will be joined to the sugar at first carbon. So this is how the DNA structure is represented. This sugar phosphate backbone, it is sugar phosphate backbone with phosphodiester bonds. Phosphate is carrying negative charge, and inside beta-N glycosidic bond joins the sugar and the nitrogenous base together. And the nucleotide, which one is nucleotide? Can you identify? I will show you. This one is the nucleotide. One nitrogenous base, one deoxyribose sugar, and one phosphate group. This is a nucleotide. So this is a repeating unit in DNA. Okay. So as you can see, it's a nucleotide, and DNA is actually like this. So thank you for viewing this video. The next video will come. I will show in the next video the different types of DNA. Okay. So till then, bye bye. You can pause this video and see the diagram clearly.